Morning students, welcome back to lecture 13. In lecture 12, in last class, we discussed about two concepts on C-value paradox, C-value enigma, what is the C-value of the genomes in terms of genome size and coding and non-coding. Further followed by that, we spoken about a little bit of more on the repetitive elements given a genome, what are the non-coding DNA leaving the coding sequences. We discussed into split the class into two halves and we discussed both the concept. Now we are coming to another important concept, very important concept in terms of the genome mapping techniques. How do you map a given genome and what are the techniques do you use to do that? The techniques are basically divided into genetic maps and physical maps. How do you construct a genetic map of a given genome? How do you construct a physical map of a given genome? There are many strategies based on some of the landmarks in the given genome sequence you can use. The strategies in detail we will talk in the next further classes, but an outline will be given here in a general introduction, different terminology used for genetic and physical maps that we will see in the present class. So this class is nothing but we are talking about the whole unit is about structural genomics. In terms of structural genomics in the introductory classes, we have spoken about in terms of the genome organization, what the genome constitutes in terms of the genes and non-coding, and what are the proteins coded by the genome and what are the structure of the proteins. In combined, we call it as structural genomics. But coming on to specific to the genome, it is called as a combination of genetic mapping, physical mapping, and the sequences here. So you are looking for a genome that is organized into different chromosomes. Of the given chromosome here, you are looking at the left, a genetic map. You are looking at the right, a physical map. Genetic map is nothing but arbitrary units of the location of the genes that is coded in a given location of the chromosome in the order. And physical map is nothing but what the corresponding sequence assembly you have done. If you have done given the genome assembly, from the sequence assembly, what are the contigs that correspond to specific location of the genes that match to the genetic map? If it matches to the contig, then what is the size of the, what is in terms of the proportion of the gene or genome in terms of length, in terms of constitution of the nucleic acid. That is what physical map, genetic map, genome in terms of chromosomes. You're looking at the end destination of the DNA sequence. So the concept in terms of the outcomes for the present class, we look into following concepts, several concepts, but some of them are the middle or Mendelian genetics that I will quickly brush up. I won't spend that much time on these two concepts. First thing we want to explain why a map is an important aid to genome sequencing or genome mapping a genome. What is really how does it help in terms of looking into the genome, in terms of sequencing the genome? And then one more important distinction we have to do that between what is a genetic map and the physical map. Are you talking about in terms of a genome map, in terms of genetics and physical? If then the map constituted by these two is completely same order of the nucleotides, or each map will have their own limitations or not? What kind of techniques you apply in terms of analyzing these and making these maps and analyzing these maps that we will see in terms briefly. And then describe the different types of marker used to construct genetic maps. What are the markers that help to fasten the construction of the genetic map? Of genetic maps, as I said, physical maps using the contig assembly. Genetic map is basically using nothing but Mendelian genetics, linkage studies, crossing over studies. Based on those studies only, a genetic map can be built. But physical map, independent of the genetics, given alleles or allele association or linkage studies, you can blindly go and look in the contig, say that this gene corresponds to this low contig is that. So to construct genetic maps, you have to go again fundamental. What is the linkage principle? What is Mendel-Morgan's principle? How do you construct a linkage map of a given two alleles? What are the homozygous, heterozygous alleles, crossing over events? So we will see in that, summarize the principle of inheritance briefly into linkage analysis. Then you go and talk a little bit about how a linkage analysis help you to make genetic maps for a given genome, or by doing so, what do you conclude from that, what it will help in terms of humans or other organisms. And then state the limitations of genetic mapping. Is it good to go just blindly with genetic mapping? Don't need to go with the physical map. Do the linkage studies and say that your disease associated with cancer is present on chromosome 9. That's it. I'm done. Or is it help you to cure the disease or not? We will look in terms of the limitations, and then further, what is the advantage of physical maps of the genome and how they are superior over genetic maps, and then how do you construct them. That will be the conclusion and a comparison of these two maps in 1 or 2 slides. So coming on to general analogy, what is a map? Why are we talking about a lot of maps here in terms of the genome? Look into a map in general terms. A map is nothing but in a given location, given a country, given in the world, it gives a specific portion, location of the country here, probably Mexico here, and then it tells you specific directions from point 1 to point 2, and then it tells that you start from Mexico here, you can go back to the city Galveston here, follow the map here, go into these kind of landmarks, take right and left, you can reach the destination B. And given a map, there will be a kind of map constitutes symbols. There will be symbols, a symbol for a water location, symbol for a garden, symbol for a park, symbol for in terms of Google map, restaurant and petrol bunk. A map will constitute of different symbols. And given the location of the map, given the symbols, if an unknown region of a given map, comparatively looking at the landmarks and given symbols, you can find unknown locations also. Given the blanks here, I have given you the landmarks of the map here, and I have given you the blanks in unknown location in the map. I want to try to match it with that, whether it's a river, whether it's a garden, or whether it's a water. So basically, the map constitute different locations, map symbols, landmarks that helps into that. And comparatively, this map is a route map. It is a kind of 3-dimensional. It will have directionality, latitude, longitude. But if you want to compare this map with the genome map, how does a genome map look? Nothing but a genome map looks in simplicity. It looks like that. You're looking at the genetic map here. Again, I've said genome map constitutes genetic map and physical map. If you look at a genetic map, it's nothing but relative position of the given genes. It can tell in terms of the alleles. And physical map, it tells a kind of specific landmarks and also specific precise location of the DNA sequences within the genes. And then you go into deducing the sequence here, going into the context that will help here. If you go from down, it will help in only physical mapping. From this point, it will not help the context sequence. This is independently looking into terms of alleles and crossover frequencies that we will see into that. So I will recapitulate again whatever we have spoken in the few slides. Genome mapping is used to identify and record the location of genes and the distances between genes on a chromosome also. You can, given the two maps here, you can precisely tell how this gene is far from this gene is present far away, or this gene is closely located in that. You can do that, and it's nothing but a genome map helps scientists to navigate around the genome. Like road maps and familiar maps, a genome map is a set of landmarks that tells people where they are in a given genome. And if they want to go into another destination in the genome, it will help them to guide and then go to the reach destination in terms of genome map also. The landmarks on a genome or map, genome map, they may be what are the landmarks in terms of the parks, petrol bunk in a given map. What are the landmarks on a genome? What we are talking about here in terms, the landmarks may be a short DNA sequences that we discussed, non-coding repetitive sequences, different types of satellites, micro and macro satellites, mini and micro satellites that may be a part of the landmark of a given genome. They may be also consensus sequences in terms of the promoter region, regulatory sites where they turn genes on and off, or they may be genes themselves. Given a location, a gene can be there associated. They can be used as a landmarks. Often genome maps help scientists to find new genes. Why I'm talking about the new genes here in terms of association with genome maps? Because when in terms of the humans or in terms of some organisms, genome is blindly you isolate given a sequences. You take how many nucleotides it is starting from the 0 in humans, starting from the nucleotide 1. you go with the nucleotide 3.2 billion base pairs, nucleotide 3.2 billion here. When you look at that, you blindly look into ATGCs only. But if you put landmarks in the genome, given a location of 1 million, 2 million, 3 million, the landmarks will tell you. And if the landmark is a known gene that you already identified, it will tell you to map other unknown genes in a given locus. Basically, you are saying that when North America was not discovered, Europeans came back and they tried to discover in this new territory what is the North America about, where they started in terms of landing in a boat. From that point, how they constructed the whole North American map coming from New York to Pennsylvania and then further. How they did it that? Because when they started, it's a new territory, they don't know anything about it. They started step by step building the whole map of North America. Same way, if in a given genome map, if unknown location, you start with small steps, you locate small landmarks, you go up to construct the whole genome map. Again, saying that if you don't have enough landmarks on the genome, you cannot build a proper genetic map again. That is a weakness. You have to drawback of the genetic map you need to look. If you don't have enough landmarks, you may not navigate properly. If you have well-established map with a well-established landmarks, that may take time, then the genetic map will be reliable. We will see one of the concepts further. What does the genome map look like? As we already seen, most everyday maps, length, width, longitudinal, like the world around us. But the genome map is not three-dimensional. It is a one-dimensional, just a linear as such, as stated. 1 nucleotide to 3.2 billion nucleotide, a full stretch of the book. Like that only, the genome map looks similar to that only, but it will give a specific distinction of chromosome 1 to 22, sex chromosomes. On the chromosome, what are the genes located? They use specifically depending on the organism, depending on the type of the gene they are talking. They use a specific letters and numbers randomly. This number associated with the gene D14S72. This associated with the transcription factor specifically GATA, and it has a specific number again. You go with giving specific notations in the genome, and it's a linear, one-dimensional, straight line with landmarks. Again, the landmarks are not regular. They are irregular intervals. You can find it. Not like you go quickly within a distance of the genome. Yes, from 1 million base pairs, I will have 10 landmarks. Continue with the 10 landmarks in the whole stretch of the genome. That is not the case. In certain position of the genome, for example, in chromosome 10, you may have less landmarks. In chromosome 1, you may have more landmarks. So how the genome maps looks like in terms of a specific, and what does it help for, how the genome maps used for. It is a genome of map, one of the genetic map you are looking for. It's looking for a chromosome 7. You are looking in terms of short and long arm of the chromosome. You are looking for a specific gene that's called cystic fibrosis gene, and the people have mapped the gene into a specific location of the chromosome. How did they map? We will see in further. But what will it will help for? By knowing the location of the gene here, cystic fibrosis, what the genome maps are used for? The genome maps help scientists find genes, particularly those involved in human disease. When you're talking about humans, you talk about disease association of a specific gene. This process is much like a game. When you're touching a gene territory, it is maybe a kind of cold and hot. In terms of cold means no association with the disease, both the patient and as a scientist, you are safer in terms of happiness. But if it's associated with a specific cancer gene, it's kind of a hot topic, hot location, and you and your patient, if you are a geneticist, you are a doctor who is working on personalized medicine, you need to work through a process to not to associate or to make the patient out of the disease. That's why how the scientists study many families affected by a disease. To locate the genetic map, as you common sense, you look in terms of the Mendelian genetics, you need a lot of recombination frequencies, a lot of crossovers, a lot of alleles you need, that we will see further briefly. So scientists study many families affected by a disease, and then based on the pedigree analysis, based on the inheritance, they will put a specific location for the gene, given the landmark, given the chromosome, and that landmark becomes specifically a landmark for the cystic fibrosis is this one, given the chromosome 7. This landmark becomes eventually a marker to associate with another new person, or the new family comes and you want to see the association with the disease or not. In other terms, we are talking about pedigree analysis. Basically, you are looking for a landmark of a gene associated with some disease, and there are two alleles in terms of M1 allele. The marker is M, but the alleles coming from the marker from father and mother are different. This is father, this is mother. Mother alleles M1, M2 of the marker, father alleles M3, M4, because mother is coming from maternal, father is coming from different parents, different alleles will come. But if a generation, given generation coming from this parent generation, the allele association will be there through genetics. Allele from two alleles will go separately. There is 4 combination. You get a combination of that. By doing so, if a mother allele is associated with the disease here, for example, an affected female, already made red, non-affected male, as per the disease symptoms, phenotypically, he is not affected, but mother is affected. Then from this progeny, you look into your combination, you look in terms of the males and females, then you can see that whatever the males coming from the allele from the mother, they are associated with the disease. By looking at this pedigree analysis, looking at this marker chromosome association with the given disease, you can detect unknown grandparent allele also. Because it's coming from the mother, by looking at this marker association, you can assume and you can hypothesis one of the grandparent, one of the mother. It's going through from mother to mother, mother only. It's going from allele associated with the mother, mother and grandmother. Based on that, you can associate a disease. So resurrection of maternal grandmother alleles in terms of the allele is M1. This may be other allele independent, but M1 is coming to this mother, from mother is in this association. That you can say, detect a hypothesis based on the marker. You deduce a disease condition, affected males, affected females, and the dead situation. If the grandparents are not available also, you can presume the allele association. By doing so, you can help, you can develop personalized medicine, you can build a marker associated with a genetic map. Again, the limitation here, you see that you are looking at how many people? 2, and then 6. You are looking at 10 people only. A combination of 10 in permutation and combination in Mendelian genetics, 10 is a very low number. You usually, in terms of the Mendel, when he studied pea plants, he was going into a combination of 16,000, 18,000 combination and making the recombination frequencies. But coming on to genetic maps in humans, limited recombinations will come. That is one of the drawback of genetic map. That's why we go with physical maps. We will see further. So this is schematic diagram of, in terms of looking at the landmarks, what I am talking. We are specifically looking at a chromosome 22 in humans using a genome sequence databases. And then the one portion here in the square, red, where the zoomed in location of the chromosome, given the location here. When you zoom into the location, it goes into this box that has a rectangle here. And there is association of specific transcription factors in a given locus here. 3 transcription factors, MYC, POL2, CTCF. These are the transcription factor associated with this location in the landmark. You can use them as a marker for a genetic map. But again, see that this transcription factor given associated with only one locus or loci, you're looking only one transcription factor. It may be binding on multiple chromosome 1, 10, 22, 23. This may be randomly binding. But as we studied combinator control of transcription factor, to switch on one gene, 3 proteins should come, 3 transcription factor and come in a protein complex, they associate. If all 3 are associated, this association may be a landmark for this position only. It may associate with other protein and transcription factor in other landmark. By looking at that also, you can genuinely go into, confidently go into this landmark location of the given genetic map. Same way, humans scientists have built in terms of ovarian cancer. They have built a specific landmarks on the chromosome, that is 3, short or long arm of the chromosome. You look into that. They have specific genes or proteins associated with that, and they located everything and they made a landmarks and the different designation in terms of the genes, location in terms of the contact. This is physical mapping. But if any disease association comes with ovarian cancer, a new patient, new territory, they don't know anything, the scientist or the practitioner will compare with the landmarks, try to do some genetic association studies to see that whether the patient is associated with ovarian cancer or not. So coming on to what are the markers that help in genetic mapping, we briefly spoke about transcription factors as landmarks, but you can again go back into specific that DNA markers, many features of the mapped that may be used in terms of the genome taken. That one concept is called restriction fragment length polymorphism, RFLPs. They can be used as a landmark for genetic mapping, and simple sequence length polymorphism, SSLPs. They can also be used. They can be divided into mini satellites and micro satellites. Mini satellites 25 base length, micro satellites little bit more shorter. You look into that from the last class. Or else you can use single nucleotide polymorphism, though they are one substitutions or point mutations only. They are highly useful in terms of differentiating polymorphism between two populations or two individuals. They are highly beneficial also to locate the genetic mapping of a given disease condition. In more in-depth detail, if you are taking a condition of the disease that's called cystic fibrosis, and this is the study started from that. For example, it's a fatal disease. The patient will die eventually if they get both alleles of the mutated alleles from parents. It's a dominant becomes and they may die. So in general, 1 in 25 Caucasians, a specific population, they have this disease. 1 in 25, high probability. The children who inherit faulty genes from both parents, maternal and paternal, they become sick. Eventually, they will die. And the best hope is to find the gene responsible for this disease, the genetic map. Based on that, what is the gene codes for in terms of the protein, you can find a target for the disease. It started in mid-80s. No one knows what are the genes that control. They slowly went step by step. They used RFLP study to identify, and they used it to construct a genetic map of the human genome. And they used one marker every 10 million nucleotides. See that. The genetic marker, you may see in the graphical representation that they are very close. But when you zoom in terms of looking into terms of genome, in terms of the nucleotides, there is only one marker every 10 million nucleotides, because our chromosomes are huge. They are shortened form of the DNA, and they constitute a longer lengths if you stretch them. Based on the RFLPs, based on the populations, they developed a map, a genetic map, and then a statistical study of 21 families again. They did not go with one family because you are using genetics, you are using Mendel, you need a lot of recombination frequency calculation. They went through 21 families that led them to area of 1 million base pairs and chromosome 7, that's specifically between two markers. They were able to locate between two markers the gene associated with cystic fibrosis, a length of 1 million base pairs. That associate with phenotypes, crossover, you give a disease and a non-disease, heterozygous, homozygous condition. There you go, so you location that. The scientist, you are happy, bingo, the patient is happy, and you could cure cystic fibrosis by studying this location. That's one of the advantages of genetic maps. So I have made few slides to understand a little bit more on, because the genetic map build on the pedigree, build on the recombination frequencies. If you have forgot your basics of genetics, I will brush up. I may not take much time on the coming few slides, 10 slides or so. I will quickly recap you in terms of the knowledge of genetics so that we can better understand what the genetic map we are talking about. So DNA, nothing but blueprint that codes and determines characteristic of an organism. The characteristic will be physical in terms of phenotype, behavioral, physiological features that controlled by the DNA. And DNA is packed into chromosomes, and a part of the DNA is called gene. Gene controls a specific trait. It is part of the DNA. That means it constitutes some nucleotide length. The position of a gene on a chromosome is called locus. And if multiple genes you are talking, it's called as a loci. And gene is heritable factor. It moves from generation to generation, and it moves through a position of crossing over. That in formation of meiosis, gametes will form, and it specific position on the chromosome. That in terms of the DNA molecule. This is but heritable factor, and that is nothing but a genetic map helps in looking at heredity. Specific position, nothing but looking in the context of the sequence assembly. You look in terms of the physical position. This is genetic map. This is physical map. Excuse me. Again, taking a loci on a chromosome, two genes are here, gene 1, gene 2. Gene 1 controls eye, gene 2 controls hair color. Each gene has a different trait to control, different location in terms of the chromosome. Genetic mapping is based on use of genetic techniques to construct maps showing the position of genes and other sequence features on a genome. Genetic techniques that usually use nothing but crossbreeding experiments in terms of lower organism given a population. Given ethical consideration, you can do crossbreeding, but in humans, you cannot do crossbreeding studies, but you go with in terms of family histories, nothing but. Physical mapping is nothing but using in terms of blindly going into molecular biology techniques. You go blindly in vitro, examine the DNA molecules in terms of physical length, in terms of base space, ATGCs, what they constitute, and then order them through assembly of a software using the genome assembly. That's physical mapping. The gene or genome mapping is describes the method to identify the locus of a gene and the distance between the genes, how closely they are in a given chromosome or given genome. Thus, the essence of all or whole genome mapping is nothing but to place a collection of molecular markers on the respective positions on the genome and detect what are the genes located based on the molecular markers. Genetic physical map, again recap. Genetic map nothing but it's the distances between the genes. And how do you measure the distances? You go in terms of Mendelian genetics. You look into a specific phenomena of linkage. Based on the linkage, based on the recombination frequencies and crossing over, you find the distance of the genetic map. Physical maps nothing but blindly go into the base space. Actually, in the genetic marker, there is a, we already saw some small drawbacks as discussed previously. Depending on the markers, depending on the availability of the lineages, numbers, may be tough. But on the physical map, they provide the accurate distances in presentation of the genome. You look a little bit more here. Gene loci are said to be linked if they are on the same chromosome. Again, going back to Mendelian genetics, independent assortment, recombination that happens in meiosis 1. You look back into that. Again, linked genes, unlinked genes. Linked genes, you see that genes A and B on the chromosome, they are present on the same chromosome, suggesting they are linked depending on the distance. When the linked genes are there, given the chromosome, homozygous, heterozygous, they always assort together of assortment. Then they go into a combination of small monohybrid pattern. You always get parental heterozygous, parent phenotype, recessive dominant alleles. But if unlinked genes, you get dihybrid, you get 4 potential gamete. Your C, the B is here, A is located in another chromosome. 2 gametes here only, 4 gametes basically, dihybrid cross, different combination. You get a recombinance here. You don't get recombinance here. If they are linked, you get unlinked recombinations here, or depending on the distance. Go in terms of crossing over, gene linkage, look into that details again, brush up your Mendelian genetics, how the crossing over will happen during meiosis. There is a crossing over of chromosomal segment chiasma, chiasma formation happens. Then you basically get into parent alleles, here homologs, parent chromosomes, synapsis happens, chiasma formation. You see that small b has been copied into here, a color change here, crossing over happens. You get a different combination of alleles here, the products from this crossover, normal parental, recombinant here. So you look into that, recombination frequencies are important to associate with the genes. And one more important, how do you say 2 genes are linked or unlinked? You do a test cross, the test cross helps you that, how the linked or not. What is the test cross? You always take a heterozygous phenotype, heterozygous in terms of 2 alleles, a combination of BB, capital B, small b, capital H, small h. You have to take an heterozygous phenotype only. The parent should be heterozygous for both alleles. Then you take a recessive allele for both, homozygous recessive. You cross into that, you get a Mendelian phenotype, 1:1:1:1. You are unlinked, the genes are unlinked. You get recombinance, suggesting that they are linked here. The traits are linked. So it's brushing. Why I'm talking about a lot of Mendelian here. It's again, you go in terms of the Thomas Hunt Morgan experiments, looking in terms of the Drosophila, different characters of a given wild type. the length and long of aristae, long legs, gray body, normal wings, and red eyes. These are the phenotypic markers that help you associated with the genotype of a given fly. It's a wild type and it is dominant, normal condition. If the alleles are mutated, you get a recessive condition. It's a mutant phenotype. Compare the shortness of aristae here, color of the eye, wings and body, different phenotypes, mutant and wild type. You take a heterozygous wild type, cross it with homozygous mutant. If there is no crossing over, no linkage, you get parental phenotype only, no crossing over. You take some crossing over happening. You see in that this long aristae parental condition have been explicitly shown, or they have been rescued in the mutant phenotype, coming a crossing over this portion. Recombinant will be formed. Same way, one more recombinant, different color of that. You can rescue different portion of that. You can look into that. Long legs, you can see a recombination can happen. So why Morgan was doing this? Because he was doing that in terms of crossover frequencies. He, based on these frequencies, he can detected a map unit distance between the given genes and chromosome based on the recombination frequencies. He showed up that you can detect based on the values there. By doing so, he detected a map for Drosophila here, different color fragments, different phenotypes. But why I'm talking about these maps and frequencies now in terms of genome maps? Because by doing so, we come back to the pedigree we studied in the human pedigree of a disease. How do you conclude the point that it is associated with the grandmother, associated with the allele M1? How do you conclude that? M1 is the association that moved from grandparent to mother, mother to the affected population in the next generation. How did you do that? So basically, they took two hypotheses that because it's linked and it is going to the generation and generation, it is associated. They assumed that the disease associated with the marker M1 and disease M1, healthy M1. By looking in the pedigree, they divided recombination frequencies, parental and recombinant, recombinant and parental. They calculated the recombination frequencies and how they deduced with the M1 as being disease, because lower the frequency, lesser the distance between the markers and they are linked. They are far away, healthy and disease M1, M2. They are far away on the chromosome. There is a higher chance of recombination. The distance is far. You get a lot of recombinance, frequency is higher, but the frequency is less means lesser distance and they are going generation again in terms of linked manner, in terms of gamete formation. The lower the frequency, lesser the distance of the marker and it is associated and in terms of linked with the disease. That's why they deduced disease M1 is associated in this pedigree. So you need recombination frequencies to deduce in terms of the genetic maps. Again, if a trait associated with single gene loci is called monogenic trait, a trait associated with multiple genes, more than two, it is called polygenic trait. It is common in humans, polygenic inheritance. You go with multiple loci, multiple gene association. You look into 1 to 2, you look into combination, you make a Gaussian curve, you look into the combination of polygenic inheritance. Again, coming on to limitations of genetic map, the map blindly or it presumes on the recombination frequencies, it presumes distance on the given genes. If you don't have crossovers, probably by mistake or by some physical disturbance in the chromosome, crossover doesn't happen properly, your recombination frequencies are wrong. It gives a wrong location of the maps. Again, there may be some recombination hotspot in a given genome. In a position of the chromosome, recombination usually doesn't happen. Then they have a limitation in terms of the Mendelian and crossover recombinance forming. So you cannot accurately depend. So you basically go in terms of analyzing a physical mapping, basically physically looking blindly, independent of alleles, independent of crossing overs. I want to go and locate the genes. It's nothing but actual physical distances in base pairs. You go using again some sort of genomic elements, DNA elements, but basically you go in terms of construction of the libraries, in terms of the contigs, and then you go into restriction fragment fingerprinting, not polymorphism here. You go use restriction fragments, you go with fingerprinting, you go with some other techniques of EST and FISH, gene marker screening, then you look at physically in terms of cystic fibrosis. that we discussed. Basically, they were generally looking to resolve regions larger than 1 MB. They looked at the context, how they looked at. They went back into the sequence assembly from yards, this much distance. They went back to the basic of the plasmids that was constructed. Based on this 2 to 6 KB region, they were able to locate which gene of the cystic fibrosis associated there, independent of looking at the crossover frequencies. That's why, how do you build a physical maps? They can be generated by from sequence assemblies. The purpose of the map is blind. You go with the contig, you go arrange that, you go on location of the gene associated in the chromosome of the given genome. Again, the techniques used: restriction mapping, fluorescent in situ hybridization, sequence tagged site mapping. Further classes we will take from genome mapping strategies, genetic mapping strategies, physical mapping strategies. Take one at a time. We can go and explain that in more detail, but have a look at it for general understanding. So based on the map-based sequencing, basically nothing but you go with a genetic and a physical mapping combination. You put a gene or marker, identify linkage, find a co-segregating marker. You need more markers in a given location. One is always not enough. Then you build a genetic map. Then you build a YAP contig, assemble, and combination of genetic and physical maps together. You annotate the genome, you assemble the genome in detail. Given if you don't know the territory of it, you go with the simple strategy. So again, ovarian cancer, if you build a map in terms of the genomic and in terms of the physical maps, this is a genome map based on recombination frequencies. This is the physical map based on the context. The genome maps gives the recombination frequencies calculated. Given the gene, given 3P chromosome, different locations are there, 25, 26, 24, and different location, different recombination frequencies based on the markers. So U is the information that is uninformative at all. It is not useful at all. You go into that, you deduce, you take into another plans, combination, genomic map here, the location, back clones, candidate genes, the physical map, go zoom in, what is the nucleotide that codes for. So again, genetic and physical maps, there is a, in terms of euchromatin and heterochromatin, there are no crossovers in heterochromatin in given a genome. And if genes are located here, you don't get crossover frequencies. That is the limitation of the genetic. Again, sometimes based on that recombination frequencies, it always the genetic map is relative. Given a gene CHA1 and GLK1, physical map is accurate. The genetic map, there is a reverse here. The GLK is coming up, CHA is 1. That is the limited resolution in terms of not enough progeny, not enough recombinational hotspot. So these two slides will talk much about the advantages of post-genomic, pre-genomic of next-generation sequencing. You just briefly look. Pre-genome, there are less markers, less distance. You look into map saturation. In post-genomic, there is higher advantage. And also pre-genomic, 10 years it takes to genetic map. Post-genomic, it takes into 3 to 4 years or 6 years. You study into that. Map saturation is that the enough landmarks on the chromosome, higher confidence in terms of the genetic map. Okay, we'll meet next week. We'll talk much more about each individual mapping strategies. I hope